About hormones, which is what the endocrine system produces, our hormones are very potent ligands, and so they are responsible for controlling things like homeostasis, right? They're one of the major players in homeostasis physiologically. They're one of the major communication molecules that say, hey, you need to do this to tissues and different organs of the body. Hormones are produced by a number of different organs in cooperation with each other and in cooperation with the nervous system to help control our control conditions, because we know that most vertebrates, particularly terrestrial vertebrates, are regulators, right? The endocrine system is also huge in a number of behaviors that you see in vertebrate groups, particularly those associated with sexual reproduction. They help maintain secondary sex characteristics in the behaviors of mating and the differences between males and females, and also obviously support the sex organs and the production of gametes. And these hormones can differ in concentrations if you're a seasonal vertebrate and you breed during a certain time of the year, like let's say you're a spring breeder. A lot of amphibians, for example, are spring breeders in temperate zones like here in Indiana. And their hormone cycles follow the breeding season, so they'll increase when they're breeding, right? Things that drive sexual reproduction are the behaviors of the males and the females. So this is a nice image here. These are chameleons, and this is the male, and they're actually mating. And you can see the male and female in this particular species of chameleon are sexually dimorphic. So the male has these horns that he uses to court the female with and also to fight off other males, and the female lacks them. So that's not always the case. It's harder to tell newts apart, for example, but a lot of vertebrates are sexually dimorphic, and it's driven by sex hormones. So we're going to talk a lot about the physiology of how hormones work, and really it's all membrane function again, so it's nothing new. You've seen almost all of this, but there are different ways that one tissue can communicate with another one, right? And coordinate physiologic responses between groups of tissues within different organs. The fastest way to do that is through electrical signaling. All right, so the fastest way for the nervous system to communicate with another cell is by producing what we call action potentials, right? And so action potentials move at great speeds. The slowest speed of an action potential in a mammal is like 3 meters per second. And if you think about that, that's pretty fast, right? The fastest nerves communicate electrical signals at 300 meters per second. So it's like instantaneous, right? So when you step out in front of the bus, you get out of the way, right? Because your nervous system communicates that information really quickly. And so these electrical signals are based on changes in membrane potential. And we're going to spend a lot of time at the end of the course on the nervous system. That's one way we can physiologically signal or communicate between tissues or cells. Another way is through chemical signals, and we call these ligands. Okay, so ligands are secreted into the extracellular fluid, right, the ECF. And they're responsible for communication within the body. It's usually one of the more common ways to do this, particularly locally. Like, you got cells within a tissue or within an organ can communicate with each other pretty easily. We just talked about an example of that, didn't we? We called it paracrine control, and how prostaglandin E controls the macula, from the macula densa, controls renin release from the granular cells, or what they call the juxtaglomerular cells. You can also pass things cytoplasmically using gap junctions, right? If cells are connected by gap junctions, one cell can communicate with another with the signal molecule by passing it through the gap junction. But target cells for ligands, or what we call targets, are any cell or tissue that has the proper receptor so that it can recognize the ligand. And in the endocrine system, we're going to focus on hormones, right, which are what we call long-distance ligands typically. And if you're a target for a hormone, then you're going to express the receptor, right, the hormone receptor. So if you can respond to insulin, you have the insulin receptor, all right? Here's an example of how the endocrine system works. Up here, it's just a great picture. You have a cell here that produces a hormone. It's going to dump it into the extracellular space. It enters the blood supply, travels long distances to its target cell, and the target cell will be the cell that has the proper receptor, right? So what that means is not all cells have a receptor, right, for particular hormones. So the insulin receptor tends to be a systemic receptor, believe it or not. Most cells have insulin receptors, so they know when glucose is available in the bloodstream. So receptors are integral proteins. They have specific three-dimensional shape. And on the extracellular side, typically, if they're in the membrane, they will fit the ligand like a lock and key. And it's not surprising that most hormones have very nice three-dimensional shape, right? And that three-dimensional shape is very unique, and they will only bind to one type of receptor. And so what this shows you is just the amino acid polypeptide sequence of a receptor. Here's your amino end of your polypeptide. Here's your carboxyl end. And there are obviously parts of the receptor that have to span the cell membrane. They have to be hydrophobic. And you can imagine these loops in this long chain here form a receptor site that would be specific for a particular type of ligand. Now over here, I put a table in from the text that gives you some major vocabulary, and I, typically would assume you know what these vocabulary words mean, right? And have found that on tests students don't. So even though I thought they did. So these are very specific to the endocrine system, and you want to make sure you go through and know these terms like an agonist versus an antagonist, affinity, competition. We're going to talk a lot about them as we move through this chapter. Now, hormones have very specific receptor specificity. So each hormone has its own receptor. So if a cell A wants to respond to 5 different hormones, it's going to need 5 different receptors, right? It's going to have to produce the receptor for all of those because each hormone or ligand has very specific three-dimensional shape. So in this example here, you have a secretory cell producing your messenger, your ligand. It's square-shaped. Your only target cell is A, right? A is the only one that's going to recognize that particular ligand because the shape of the ligand fits the receptor perfectly, like a lock into a key. You all know, even if the key fits into the lock, if it's not the right lock, it won't open the door, right? It's the same thing with these hormones and receptors. It's that perfect, that as far as the way the ligand fits the receptor. So this gives you a very specific system so that cells can be really, can really control just certain tissues in the body and be very organized in the way that they do it. Now this image just reminds you of how receptors are produced. Okay, so we know if we're going to be a receptor, we are going to be delivered by a vesicle to the cell membrane, right? And so the endomembrane system is responsible for that happening. And so you're going to translate the messenger RNA for the receptor at the rough ER, right? And then you're going to use a transport vesicle to package and change that receptor in the Golgi. In the Golgi apparatus is where you actually add the receptor to the cell membrane typically. And then that means the Golgi membrane can pinch off to become a secretory vesicle. And notice now we're carrying that specific receptor. When stimulated, the cell will add that secretory vesicle via what we call exocytosis to the cell membrane, but instead of releasing, right, our product, we are just creating cell membrane here, right? We're adding the receptor to the cell membrane, as you can see it right here, right? And now I can respond to a specific hormone. So cells can add more receptors this way, or I could take the receptors away. I can endocytotically bring the receptors back into a vesicle and take them off. And so this is called upregulation when you add receptors, and downregulation when you remove receptors. Cells are very sensitive. If there's a lot of hormone and you're really speeding up their physiology and their metabolism, cells are unhappy. So they can downregulate, right? So they're not so sensitive. Does that make sense? To this ligand. If there's not a lot of ligand available, they might upregulate the receptor so that they are more sensitive. So sensitivity, if you will, for a target cell depends on kind of the number of receptors that you have on the surface. Does that make sense? All right, so let's talk about the different ways cells can communicate with one another. First way, cytoplasmic to cytoplasmic communication happens through gap junctions. So if cells are close together and express gap junctions, then they can communicate and coordinate really quickly. So when we get to the heart in our next chapter, that's the next chapter we're going to cover is the heart, we're going to have what we call networks of cardiac muscle that are all connected to one another. And if you stimulate one of them, you stimulate all of them. And the reason for that is because they have gap junctions. And it's rapid, very coordinated communication. And so it allows for direct, very rapid, coordinated features to happen between groups of cells at the same time. And so when you stimulate one cardiac muscle cell, all of them contract at the same time. So the protein here that's involved are connexins, right? Connexins are these channels, these pieces of hollow protein that span both cell membranes and allow for cytoplasmic communication. The next one is what's called contact-dependent signals, where one cell has the ligand, but it's attached to the outside of their cell membrane, and the other cell has the receptor. And in order for them to meet, they have to come in contact with one another, right? They have to touch. And this happens a lot between epithelial cells. This is how epithelial cells know, hey, we're supposed to touch, right? And they make contact with each other. This is how white blood cells communicate with one another. One white blood cell might have a receptor for a particular ligand, and the other white blood cell turns on that cell by making contact with it and having the right ligand on its surface. And that can start an immune response, for example. And so these are contact dependence where the cells actually have to come in contact with one another. And the reason they have to do this, contacting each other, is because the receptor and the ligand are in the cell membrane, right, of each cell. And the cells have to come into close proximity to each other. Now, the next type is cell communication is going to deal with receptors being expressed and then we releasing a ligand that's mobile into the extracellular fluid. All right? And the first type of signal that can be released and then find its receptor is what's called an autocrine signal. An autocrine signal is when a cell produces a ligand and it also expresses the receptor for that ligand. And so the ligand binds to itself and causes the cell to do something. All right? Great example of this. When a cell is infected by a virus, if it can get some proteins produced before the virus takes over its cellular machinery, it will produce a kill signal, all right, that has receptors on the surface of that cell that cause the cell to die, all right? It's basically, suicide for the rest of the health of the tissues or cells around it, all right? And so these signals are signals that are often involved with inflammation, with immunity, with infection. At the same time, this cell might send out some local signals to the cells around it saying, hey, I'm infected with the virus, produce antivirals, right? And so those are what we call paracrine signals, which is the next type of ligand binding signal, where it's paracrine. I always like to think paracrine is local signals that only happen within a specific tissue and nowhere else in the body, where the cells of a tissue are communicating with one another and nobody else. And so this is when one cell produces a ligand that diffuses just around to the adjacent cells and changes their physiology. Make sense, the difference between autocrine and paracrine? And then what this chapter really focuses on are endocrine signals. Signals which are released by an endocrine cell or gland called a hormone. The hormone travels through the extracellular space, enters the bloodstream, and then travels long distances away from the endocrine source and delivers hormones to target cells, those that have the proper receptor. Right? And there are lots of different hormones that are involved with controlling physiology and homeostasis within a vertebrate. We're going to learn about some of them, not all of them. We're going to, again, take that theme-based approach where we're going to learn how hormones work, and then I'm going to give you a few examples. Right? I'm not going to make you learn every hormone in the vertebrate body, but I'm going to give you examples of hormones that I think are important to understand and kind of how they work. One other group of molecules that if I'm doing animal phys that I got to talk about are what are called pheromones. All right, a pheromone is a hormone released into the environment, not into the extracellular space, and will communicate with other individuals of the same species. So, hormones often attract mates. This is one way a lot of organisms attract mates, particularly arthropods and fish. So here's a female moth, here is a male moth. The male moths usually are the ones with the big, huge antennae. And what those antennae pick up are pheromones, right? So that they can find the females. And so the male will follow the little chemical cue that she produces. And she usually sits around and waits or, on a plant or whatnot until the male finds her, and then they can mate and reproduce. Here's a great example of a powerful pheromone that's produced by silkworms. It's called bombycol. And we have actually, as humans, figured out what bombycol looks like, and we've actually mass produced it. And you can put it on sticky paper and attract all the males anywhere near your trees to prevent silkworms from damaging your trees, because the caterpillars build those big silk webs around, you've seen them in the late summer, around trees, and they destroy trees, particularly if they're in infestation. They can kill a tree, and this is one way to deal with them. This is also how Japanese beetles traps work. They have pheromones in them that attract the beetles. It's a better, more environmentally safe way to destroy pests without spraying pesticides over everything, right? Which have dramatic effects on lots of organisms. So that's what pheromones are. Okay, so hormones. Hormones allow for cell-to-cell communication. They're chemical signals. They're secreted by cells, and it shouldn't be very surprising that they're also usually have quite dramatic three-dimensional structures. The structures are not only ringed, which gives them good three-dimensional structure to make a receptor, but they're also very stable often, right? So that they last a long time. And they are transported through the blood and will affect target tissues that have the proper receptor. So here are examples of steroids. Right, steroids are a class of hormone that are cholesterol derivatives, and all that I'm going to do to the cholesterol molecule is use the ringed backbone here, your 4 rings, and then just change the R groups and where the double bonds are found. And that gives you dramatic changes in three-dimensional shape so that they fit specific receptors. And so, for example, it's pretty amazing. Look at testosterone and look at estradiol. Male hormone, female hormone. And if you look at them, holy moly, they look very similar, right? But they're very different in how they can bind the receptor. Most females produce testosterone first in the ovary and convert it to estrogen. Males produce estrogen too, even human males, right? And it's important in certain parts of the reproductive tract. So male birds, songbirds, produce estrogen because estrogen is responsible for the beautiful song that they produce, not testosterone, right? The male hormone, the dumb hormone, right? The hormone that makes us males, right? I say me because there are no other males in here. So I can say that. Estrogen, make you smart. Smart hormone. At least that's what my wife tells me. She's smart.
She's a pharmacist.
She is. She's very bright. I concur. Okay, two important characteristics of all hormones. You kind of heard me talk about one of them already. That's specificity. Their three-dimensional shape makes them very nicely tailored to fit one type of receptor. And if I want to recognize that hormone, I have to produce the hormone's receptor in order to do that. So it's a lock and key kind of mechanism, and the shape of the hormone is really important. The other thing is that most hormones have great affinity, or they're very potent is another way to think about it. They work in the parts per trillion in the body, which is incredible. You only need one or two or three of them in most cases making it to a target cell in order to get a huge response from that target cell. And we're going to talk about why. It's all about kinase cascades. All right? It causes this huge response in the cell because if you activate a kinase, it can start phosphorylating lots of proteins, right? And so affinity is a big thing. And cells can help with that a little bit. Your target cells, as I've already mentioned, they can become more sensitive or have higher affinity for a hormone by upregulating their receptors, adding more. Or they can have less affinity or become less sensitive by downregulating the receptor. So often having a G-coupled receptor as my hormone receptor is what allows for affinity to occur because it allows for a major response because of the activation of enzymes. And we'll talk about that magnification ability when we give some specific examples here of hormones. So let's start talking about the different types of receptors. Now, a lot of hormones are water soluble. Not all, but a lot of them are water soluble. And so if they're water soluble, they're going to have to find their receptor at the surface of the cell, right? And we know that receptors and hormones, the hormones are attaching to the outside of the receptor. That is not direct communication in the cytoplasm, right? They're going to have to activate or cause the activation of a second messenger system, right? So if a hormone comes in and binds to its receptor, it's going to change the shape of the receptor because remember, proteins have wiggle. When they bind something, they tend to change shape a little bit. And that will activate something on the cytoplasmic side of the membrane, like a G protein or an enzyme, or maybe the receptor has a built-in enzyme already part of its structure. Like in this example, this is a tyrosine kinase receptor. That's what the TK stands for. When the hormone comes in and binds to the receptor, it changes the shape of the receptor, turning on the tyrosine kinase, which can then phosphorylate things because it's a kinase, right? And so it starts phosphorylating proteins on the cytoplasmic side. Or I could activate an enzyme like adenylate cyclase, that's what AE stands for here, and produce a second messenger like CAMP, right? Those activate kinases here and start causing phosphorylation of proteins, which causes cellular responses. That's how most hormones work. So it's not surprising. You remember that cell communication figure I showed you a long time ago? With guanylate cyclase and adenylate cyclase and phospholipase C. Those are the major second messenger systems that we're going to utilize. So if you remember those, it'll be easy, right? Or hopefully you remember enough of it that if you review, it's not too bad. Now, probably the easiest, or I shouldn't say easiest, the most simple setup of a receptor is when the receptor is also a channel. All right, it's a gated channel. And so when the first messenger comes in and binds to the receptor, the channel opens or closes sometimes, right? It changes the shape of the receptor, opening or closing the channel, and then things can escape, all right, or move. Like, so for example, if this is a gated receptor that is a calcium channel, when the first messenger comes in and binds, it opens, allowing calcium into the cell, and we know calcium is a very potent second messenger, isn't it? Right, and this is one way that we can make a real simple system and have calcium come in here, bind to, say, comodulin, and then activate calcium-dependent kinases, and you can start a whole enzyme cascade or change in physiology. But overall, most receptors are complicated, not because we did this on purpose, right, to make students have to learn it, but because this is what allows for affinity or potency, all right? That is, this allows a cell to respond quickly and in very great power of physiology, if you will, in response to one or two of the first messengers actually making it to our target cell. And these G-coupled receptors are going to activate an enzyme typically, and that enzyme is going to produce a second messenger that's going to activate protein-dependent kinases. And we know here with CAMP and adenylate cyclase that this first protein kinase is protein kinase A, right? We've mentioned that several times over the semester, which then will phosphorylate other proteins, and you get a huge response to one messenger. So CAMP is a very popular way to do this. Guanylate cyclase is a very popular way to do this. IP3 and DAG is a very, very popular way to do this, right? I'm not going to bore you again with how this works. You already know, right? And this is how you get intracellular calcium release, right? Is through IP3 and the smooth endoplasmic reticulum, which has IP3 calcium receptors, which allows calcium to enter the cytoplasm. So we're going to talk about a number of different hormones that have these different types of setups. Now, calcium is a popular second messenger in the endocrine system and in cell-to-cell communication. And comodulin is often the calcium sponge within the cytoplasm that allows calcium to activate kinases. And so calcium never acts alone. I hope you figure that out. It kind of binds to something first, right? And when it binds to that, it forms a calcium complex that can then activate the proteins. And the hormone examples, calcium that comes in cytostolically, whether it's from a membrane-bound receptor or it's from an intracellular source like the ER, once calcium's in the cytoplasm, it binds to comodulin, which is simply a calcium sponge. It soaks up the calcium. And then the calcium comodulin complex will activate protein kinases, and that causes your response. Okay, last slide here. Calcium as a second messenger. So again, calcium can do a whole bunch of different things. It's a very, very potent second messenger, and it causes a ton of responses in a lot of different tissues. But almost always what it's going to do is activate some kinase, right? To turn on other inactive enzymes that are sitting around in the cytoplasm. And these cause major kinase cascades or physiological cascades that end up with one hormone causing the activation of thousands of enzymes because of phosphorylation, which changes the physiology of the cell completely. There are lots of different proteins that can bind calcium. I'll give you a couple here. Comodulin's one, troponin is another. All right, troponin's found in muscle tissue. There's caldesmin, there's synaptotangmin, there's a whole bunch of them, right? We're going to learn different names of them as we go through different chapters. All right.